Assalamu alaikum, everyone. I'm Abdulrahman Farahat. I'm a second year medical student, and our lecture today is a pretty simple one. It's about small intestinal motility and secretion. If I'm going too fast or anything, please do stop me at any time. If you have any questions, you can contact me here. So whatever you use. And okay, so let's start. I divided the lecture into four main parts. We'll start with the structure or the anatomy, which I'm pretty sure most of you already know. And then you have the function or the physiology aspect of the lecture, which will be the bulk of this session. And finally, we'll take a look at some conditions and some questions. Now, I know this lecture, as I said, it's pretty simple. So my main point here today is I want to ensure that you guys know the main or the high yield concept. And so I'll be stressing out on some topics, and please do take note of them. Yeah, you'll probably get asked about. So starting with the first thing, which is the structure or the anatomy. Okay, so everybody knows this small intestine. It's made of three parts: the duodenum, the jejunum, and the ileum. And yeah, so the duodenum would be the first part, the jejunum second, and the ileum third. There's not much you can say about them. The jejunum is slightly more larger than the ileum, and so that could be a differentiative thing between both. Or other than that, you cannot tell when the jejunum begins, when the jejunum ends, or when the ileum begins. You see. Moving on. Oh, there are certain points I need you to remember. So we'll speak about the vascular supply and the innervation. So for the vascular supply, we have the artery, which would be the superior mesenteric artery, a direct branch from the abdominal aorta, and the vein. Does anybody know what vein that would be? Okay, so that would be the superior mesenteric vein. And eventually that vein would drain into the portal vein. So this is a picture of the portal vein. And if you could imagine like a liver structure, okay, this is a liver structure right there. So try to imagine the portal vein going through. Okay, so now for the innervation, you see here you'd have the sympathetic nervous system and the parasympathetic, and the most important one would be the enteric nervous system, because it's a characteristic of the whole GI system that it's the only system in our body, the only organ system to have its own specific kind of, you could say, innervation, which is through the enteric nervous system. Now, the sympathetic nervous system, its main function, or let's speak about the nerves first. So the nerve of the sympathetic nervous system would be through the celiac nerve and the superior mesenteric nerve. And the main function of the superior mesenteric, oh, okay, and the main function of the superior mesenteric would be, or the sympathetic nervous system would be to contract, decrease the contraction in the GI tract. Now, the parasympathetic, on the other hand, you'd have the nerve, which is the vagus nerve, and the through acetylcholine, it would increase the contraction. So that's basically the structure or anatomy. There's not much to say about it, and I'm pretty sure you guys already know it. So yeah, does anybody have any problems with that? Okay, so now let's go on to the function and the mechanisms of, or the pathways found. And we'll be starting with a basic overview of the main functions of the small intestine. So when I tell you small intestine, two things should come to mind. First of all, it digests the food. So in the small intestine, we digest food. And then we absorb the nutrients and electrolytes we get from the digested products, right? So when we digest food, you might ask how. So we have carbohydrates and proteins, which would be digested through the pancreatic enzyme. You guys took the pancreatic secretions lecture, so I'm pretty sure you guys know the pancreatic enzymes. So could anybody give me an example of a pancreatic enzyme that would break down carbohydrates? Okay, so for example, let's say alpha amylase and the pancreatic enzyme for proteins, for example, trypsin. At the same time, you'd have lipids that through pancreatic lipases, you'd break down lipids through pancreatic lipases and the bile salts. So bile salts, you might they do not break down themselves, but they help in the reabsorption and formation of the micelle. Now, moving on, for the absorption, here there's something really important you guys should keep in mind. So for the jejunum, it's pretty general. There's a lot of stuff. Almost everything gets absorbed in the jejunum: electrolyte, iron, folate, fat, proteins, carbohydrate, whatever. Everything almost gets absorbed in the jejunum. The thing is, the most important thing to remember would be vitamin B12. So vitamin B12, you should know that it gets absorbed in the ileum. And I'm not sure if you guys took this condition or not, but for example, the case scenario would be like a patient comes in with a certain disease that causes inflammation in the intestines, right? And this inflammation, the only way you could treat it would be to remove that part of the small intestine. So to remove what that part is usually the ileum, so where the inflammation usually happens. So when you remove the ileum, the patient, they would say then the patient follows up after 3 months, and he presents with typical presentations of anemia, so shortness of breath. He has so yeah, typical presentation. Nothing coming up to my mind, but he has anemia. You might think he has anemia, but and he actually has, but the cause of the anemia would be due to this vitamin B12 deficiency. And that would be one of the answer choices. So the answer choices, so the question would be like, which of the following could be a cause for the patient's symptoms? And you would say a deficiency in vitamin B12. So it's that clear. You have to keep that in mind. And the disease, by the way, is Crohn's disease. Just to make, I'm not sure if you took it or not, but yeah, always keep it in mind. Is that okay with everyone? Okay. Oh, and here we have a checkpoint. If any of you have questions. Now, to the motility in the small intestine. Now, in the motility in the small intestine, we divided into two parts. The small intestine when there is no food, and the small intestine when we have food, or when we are digesting material. So when we don't have food, we are in a phase called the migrating motility complex. So this is the motility that's happening continuously throughout the small intestine. It starts from the stomach. I'm pretty sure you took it in the gastric motility lecture. So in the small intestine lecture, so it continues. It starts, its contractions, but start from the stomach, and it continues all the way to the small intestine. And it serves various functions. Like, see here, it happens every 90 minutes. It should be known, and it's mediated mainly by motilin. Okay, but the main function I want you guys to focus on, which is also another concept that I want you to keep at the back of your mind, because we're going to speak about it at the end, which is removing bacteria. So it removes shed cells, food residues, foreign bodies, and whatever. But the main thing is it pushes out or kicks out any bacterial residues that remain in the stomach, in the small intestine. It keeps on pushing it towards the large intestine. And finally, of course, once you are feeding, this would interrupt the migrating motility complex. And now you will have either one of two motility actions. Either you'll start with rhythmic segmentation phase, where in rhythmic segmentation, what happens is you can see here, there's a contraction, here's a contraction, and here's the relaxation, right? So now what would happen is a contraction would happen here, and relaxation here and here. So relaxation on both sides and contraction on the same side. This would lead to some food would propel forward, some food would propel backward. And the food would end up mixing together in the relaxed area. And so this would be the main function of the rhythmic segmentation phase. So when I tell you rhythmic segmentation phase, the first function that comes to mind would be the mixing. Mixing of what? Mixing of the bolus of food with the pancreatic enzymes, the digestive juices secreted. Everything would mix up and it would help in digestion. Now, this is the most frequent motility action happening in the small intestine. And the stimulus for this would be the introduction of liquid nutrients. So once liquid nutrients enter into the small intestine, you'd have this activation of the rhythmic segmentation phase and contraction and mixing up of material. Is the rhythmic segmentation phase done? Clear? Any problems with it?
Clear.
Okay. Now, for peristalsis. For peristalsis, it's the other motility mechanism that happens during feeding. Now, peristalsis, I'm pretty sure you guys took it even in high school. Really quick over it would be a contraction would happen behind the bolus of food, and the relaxation would happen after the bolus of food. So this, you might think of it like propelling the food forward. So it would look something like this. You would contract this part and relax the one in front of it, so the food would be pushed forward, and it would continue pushing. So the main function when you speak about peristalsis, when I tell you peristalsis, the main function, yes, so relaxation by VIP, and we'll speak about. So the main function of peristalsis would be the movement of food. So again, let's really quickly review this. What if I tell you what's the main function of the migrating motility complex? You would say
to remove shed cells and bacteria.
yeah, and remove any waste products or anything remaining undigested material. It would push it all the way toward the large intestine. Perfect. And now, what's the main function of the rhythmic segmentation phase or motility?
Mixing.
Mixing. Yeah. So mixing of the material. And finally, what's the function of the peristalsis?
It moves into the ileum, like it moves from the duodenum to the ileum, like it moves the food so that it can get more digested.
So yeah, essentially it propels the food through the small intestine. Okay, so that's it. That's the thing I'm trying to get at. It's the less frequent, the stimulus. This is also a thing to keep in mind, that the stimulus for peristalsis would be distension by bolus of chyme. So the small intestine is usually relaxed like this, and once a food or a bolus of food or chyme enters, it will distend the small intestine. And this distension is what initiates peristalsis. Of course, it's controlled by the enteric nervous system. So it's not controlled by the sympathetic or parasympathetic. Forget about them. When you speak about peristalsis, your main focus should be enteric or the enteric nervous system. Okay, so now let's speak about the mechanism. And we have our first friend right here, anger. I'm not sure if you guys watched Inside Out or not, but anger is typically an angry guy. So this guy, for me, represents one of the cells that is important in the mechanism for peristalsis. Do you guys have an idea what that cell might be? Okay, that would be the EC cell or the enterochromaffin cell. And I'm going to tell you why. Because as I told you, the small intestine is normally relaxed, nothing's happening. And then all of a sudden, a bolus of food starts entering and descends and pushing on the small intestine. And the other cells are like cool with it. It's fine. They get descended, they have no problem, except the EC cells. This guy gets angry. This is the anger. This guy, once you descend and he gets pushed, he starts the whole mechanism of action and starts attacking everybody. Once he gets angered, he releases 5-hydroxytryptamine or serotonin. Serotonin goes to the afferent neurons, stimulates the afferent neuron of the enteric nervous system. Again, peristalsis is controlled by the enteric nervous system. The afferent to the enteric would stimulate or activate the efferent or the motor neurons. And then the efferent, as you can see, they have two parts. The first part would go behind the bolus. So this is more orally. And it would go behind the bolus and release acetylcholine and substance P. And this would induce contraction. So you would contract behind the bolus. And the other part would go after the bolus or aborally, and they will release vasoactive intestinal peptide and nitric oxide. And as you said, these would cause relaxation. Perfect. So the whole problem started by distension of anger. Anger got angry and they released serotonin. Good stuff. And yeah, this is basically everything I just said. So distension of gut wall by bolus activates the EC cell or our anger. And then the EC cells will release serotonin or 5-hydroxytryptamine and stimulate afferent sensory neurons. So the sensory neuron. The afferent sensory would then go to the enteric nervous system and stimulate the efferent motor neuron. The efferent, as we said, they will divide into two parts, a part going behind the bolus and a part going after the bolus. So the ones going behind the bolus are going to contract. So we're going to release acetylcholine and substance P. and the one going after the bolus are going to relax, releasing vasoactive intestinal peptide and nitric oxide. And a good way to remember vasoactive intestinal peptide that it relaxes is through this mnemonic I used to use, which is VIP. Generally, we know VIP is a very important person. And so essentially, VIP is clearing the way for the bolus to go through the small intestine. So that's like a link you could do. Is everything clear? Hopefully, I'm not going too fast. Okay, so now for scenario time. You see, I assume that you guys watched Family Guy and you know Peter Griffin. So Peter Griffin likes to eat. He went to the convenience store and bought some food. Okay, he's enjoying his life. Then, while sitting with his family and his son, Tiwi, all of a sudden, boom, oh, oh, boom, vomiting happened. A lot of vomiting, and it didn't stop there. Continued vomiting, and it's just a terrible day for Peter. So, what is the mechanism behind vomiting? Vomiting happens due to something we call retroperistalsis. So essentially, vomiting starts with retroperistalsis. So exactly, yeah, retrograde. So first of all, let's dissect this word. Peristalsis, we already know, contraction before and relaxation after, causing movement of bolus from the oral to aborally, so forward. Now, retroperistalsis, once we add retro, this means retrograde or reverse peristalsis. So now we're contracting after the bolus and relaxing before the bolus, such that the bolus is going back into the stomach, right? Now, retroperistalsis, again, we have Peter Griffin here. There's a mechanism behind retroperistalsis. So why do we have vomiting? So vomiting happens. So let's start. Peter Griffin ate something irritating or toxic. The toxic stuff entered into the small intestine. It got digested, broken down into simple material, and then it gets absorbed. The absorbed material goes into the blood. The blood circulates all over his body and goes to the brain, a region in the brain called the vomiting center, which is found in the medulla in the brain, right? Now, the vomiting center is like a security guard. This guy looks at the blood and makes sure that nothing toxic or nothing bad is there. So once it detects like toxic material or irritating substances in the blood, the vomiting center is alarmed and it starts sending signals to the small intestine. What's it going to do? It's going to reverse the peristalsis happening in the small intestine. So the vomiting center signals the small intestine to undergo retroperistalsis and pump or like push the bolus or the toxic material back into the stomach and eventually for expulsion. Now, peristalsis would enter into the stomach and kind of stop. The food would enter there, and where does it go? What happens? How do we eject the food, right? So here, our body also helps. So first you increase or the contraction, the diaphragm would contract. Diaphragm would contract, the abdominal muscles as well, and this would increase the intra-abdominal pressure. And by increasing the intra-abdominal pressure, it's like you're squeezing on the stomach and you're increasing the intragastric pressure. And then once that pressure is high enough to overwhelm that of the lower esophageal sphincter, the food would then pass up into the esophagus. Now, you're not done there because still you have, if you know, the esophagus, it has an upper esophageal sphincter and a lower esophageal sphincter. So you pump blood, or not blood, you pump the food, you're pushing it into the esophagus, you pass the first checkpoint, now you have another checkpoint. How are you going to pass that? It's not in the abdomen, it's in the thorax. So we're going to increase the intrathoracic pressure. Again, how's that done? Through the diaphragm and the intercostal muscles. They are going to contract and overwhelm the esophagus such that they open the upper esophageal sphincter and eventually the food would come out of your mouth and as emesis or vomit. So is the mechanism clear to everyone? Okay. Yes, yes. Retching would happen if, yeah, so exactly as you said, if there is pressure if you cannot overwhelm the pressure of the upper esophageal sphincter, and the food would remain in the esophagus, and you might feel it, يعني, food stuck in the esophagus. You could feel like opening of the lower esophageal sphincter, and this could also lead to heartburn or whatever. Right. So yeah, that's stretching. Perfect. Okay, now the ileocecal junction. The ileocecal junction, the thing I need you guys to focus on is its function. But first, let's look at its structure. So the ileocecal junction is made up of two things, the ileocecal sphincter and the iliac papilla. And essentially, what both of them do is they push, or like they produce a tonic contraction over that region, which is the ileum and cecum between them, and preventing stuff from going in both directions, by the way. So prevent stuff from the ileum protruding into the cecum and going back. However, the ileum could surpass this pressure through contractions and ability, but the cecum does not have that ability to retrograde. So in normal conditions, the ileocecal junction would, if you think about its function, its main function would be to prevent retrograde of material from the cecum, or food from the cecum back into the small intestine, right? So that's the main function of the ileocecal. Another function, as you mentioned, its major function is to prevent small intestinal bacterial overgrowth. So yeah, that's one of its important functions as well. So mainly, it would prevent reflux of the material from colon back to ileum, but also, you're right, I wrote it here, prevent small intestinal bacterial overgrowth. So normally in the large intestine, you would have a bacterial environment there, which we call the normal microbiota of the small intestine, of the large intestine. And so this bacteria is important for us for protective mechanisms and whatever other functions there is. However, so the ileocecal junction would prevent those bacteria from the large intestine from entering or escaping the large intestine and entering into the small intestine. Because once they enter into the small intestine, we're going to have another. But let's keep it at this for now, because we're done with the physiology part of everything. We're going to speak about the conditions now. So let's make sure that everybody's clear with the physiology mechanisms and everything, right?
Clear.
Okay. Now the conditions or diseases. So the first thing, we'll speak about the small intestinal bacterial overgrowth. And this would happen, as you said, you'd have the bacteria in the large intestine would overgrow into the small intestine. And you have multiple reasons that would happen. We already spoke about this one, which is the loss or dysfunction of the ileocecal junction. So if the ileocecal junction is not there, or it's weak, you have paralysis of some sort of the muscles there. This would allow the large intestinal bacteria to retrograde back into the small intestine, causing small intestinal bacteria overgrowth. They would overgrow in the small intestine. Now, there are two other reasons that might happen. First of all, you might have decreased gastric acid. So, subhanAllah, our stomach has hydrochloric acid. And it's actually one of our immune mechanisms against foreign bodies or organisms in general. So if you eat something that has bacteria, yes, bacteria could escape, but most of the time it won't. Why? Because it would enter into the stomach, and it would get degraded and destroyed by the hydrochloric acid present. So if a patient or a person is taking proton pump inhibitors, so a proton pump inhibitor essentially prevents protons from being pumped into the stomach and prevents hydrochloric acid from forming. So there is no acid anymore. So the bacteria would enter, it would never get degraded, and it would escape and enter into the small intestine. So that's a reason for a small intestinal bacterial overgrowth. Now, the other thing is decreased motility. Now, decreased motility could be caused by two things. For example, the intestinal obstruction, where, for example, you might have an intestine twist, a twist in the small intestine, and this will prevent any material from moving. There is no motility. This would also cause small intestinal bacterial overgrowth. But also, if you remember, we spoke about the migrating motility complex. So the migrating motility complex, we said one of its main functions is to remove everything, including bacteria. So the migrating motility complex would just kick out everything from the stomach, from the small intestine into the large intestine. It's not important. Kick it out into the large intestine. So that's so if a person has a problem with the migrating motility complex, it would also lead to decreased motility and small intestinal bacterial overgrowth. So these are all causes, and for every cause, we have an effect. So what's the effect of small intestinal bacterial overgrowth? Two main things, two main presentations would happen, or symptoms would happen to a patient with small intestinal bacterial overgrowth. First thing would be cytorrhea, and the second thing would be malnutrition. Do any of you know what cytorrhea is? Okay, so steatorrhea would refer to having a lot of fats and lipids, undigested free fatty acids in your stool or feces. So when you have a lot of these stuff in your stool or feces, we refer to this as steatorrhea. And malnutrition, I'm pretty sure you guys know what malnutrition is. So the patient or a person is malnourished, he doesn't have enough vitamins, enough nutrients, carbohydrates, proteins, they are all depleted. So that's a malnourished person. Now, why does this happen? Why do you think small intestinal bacterial overgrowth causes steatorrhea and malnutrition? So I brought this photo from the or picture from the slides, and honestly, it's pretty concise and clear. So first, at the top, you could see a normal intestine, right? And here's the colon, and there's no bacteria in the ileum. Everything is perfect, nothing's wrong. At the bottom here, you would see small intestinal bacterial overgrowth. There is invasion of the bacteria. So at the colon, you can see the ileocecal junction for any reason or another, it's no longer functional. And so the bacteria would propel or invade the small intestine, starting by the ileum, jejunum, and whatever. So once the bacteria invade that structure, first thing, it's going to cause blockage. So it could pile up like a bacteria here, and another one here, and it continues piling up and closing over or clumping up over the microvilli. And so by preventing the microvilli, essentially preventing the absorption of anything or degrading of anything there. So that's why you might have cytorrhea, because essentially you're blocking the absorption. And also you might say you'd have malnutrition. But there's also another reason for malnutrition. Malnutrition would happen because the bacteria would start stealing the food particles you haven't digested or picked up yet. So, for example, leftover glucose and galactose here, the bacteria would start eating it up, and now it blocked the absorption and it's stealing your material. It's destroying everything. So, small intestinal bacterial overgrowth destroys everything. So, when you speak about small intestinal bacterial overgrowth, think of these two things: cytorrhea and malnutrition. Cool. So yeah, guys, this would be a topic, a question in the exam, really simple question in the exam. A patient presenting with cytorrhea and malnutrition. Once you see those two words, small intestinal bacterial overgrowth. Of course, there must be stuff added to it. For example, like he had a surgery in his ileocecal junction and he had to remove it. So yeah, connect stuff like this. Okay, let's move on. Now we have another condition which also causes maldigestion and malabsorption. So it is called gluten-sensitive enteropathy. And before we get into the whole pathophysiology of it, let's try to dissect this word a little. So gluten, some sort of way, is involved in this mechanism. Sensitive, so sensitive would tend to mean like somebody allergic or sensitive to something. Enteropathy, so entero is the GI or alimentary canal, and the pathy is pathology. So you'd have some sort of allergic response to gluten in the GI tract, right? Okay, so let's see the scenario. We have this amazing gluten protein molecule, and it enters into the small intestine. In a normal body, nothing would happen. You would degrade this gluten, you would pick it up, you would get rid of it. It's fine. Nothing would happen with this gluten protein, right? The problem happens with these patients because gluten for them is something foreign. It's a foreign material. And so once they get exposed to gluten, they start forming a lot of antibodies. And this would lead to a whole inflammatory response in the small intestine. And it's also important to note that in the small intestine, the small intestinal cells itself, they are very weak. And any inflammation there would cause destruction of everything in this cell. However, gluten-sensitive enteropathy mainly affects a certain point on the enterocytes, which would be these villi right there. So this whole inflammatory reaction that would happen in the small intestine would affect the microvilli, and it would cause something we call as celiac disease. So you see here celiac disease, and so this is a normal intestine. It has amazing, happily living microvilli, and here you have celiac disease, where the villi is no longer there, and everything is going to is a mess, right? So that's essentially a gluten-sensitive enteropathy. And this happens, as we said, it's an autoimmune response of the system to the gluten protein. Now, here there are certain symptoms that come concurrently with the celiac disease. And if you think about it, it's pretty logical to happen. So you'd have loss of surface area. So the microvilli, which are important for the surface area, you no longer have microvilli. And now you're no longer absorbing any of the electrolytes, of the glucose, of the various molecules found in the small intestine. And what's the number one rule you should never forget? When you have a lot of solutes in a certain area, water would always follow. Now you have no transport, you have less surface area, nothing, everything is remaining in the small intestine. Now, osmotic diarrhea. All of a sudden, all the water would come and it would cause osmotic diarrhea. Always remember that. Now, also, you'd have here imbalance between absorptive and secretory fluxes, less absorption, of course. So you'd no longer absorb, you'd start secreting more because of lack of the microvilli. And also the unabsorbed fatty acids. You'd have unabsorbed fatty acids. So again, you would have a case of steatorrhea. You'd have steatorrhea because the free fatty acids are going to remain and eventually they'll get excreted in the stools. Now, the last condition I'll speak about, which is the lactate deficiency. So I'm going to focus on two main signs that if you see in lactate in a person, you have to diagnose them as a lactate deficient patient. So now let's really quickly speak about a normal person. A normal person, he would drink milk normally. It has a lot of lactose. Lactose would enter into the small intestine. You have an enzyme, lactate dehydrogenase, or lactose dehydrogenase. And this enzyme is going to break down lactose into galactose and glucose. Now, galactose and glucose are simple monomers, or monosaccharides, and you're going to absorb them easily. Now, this is a normal person, right? A patient who has lactate deficiency lacks this enzyme, lacks lactate dehydrogenase. And lactose is a disaccharide and it cannot easily be absorbed, right? So now, lactose would remain in the small intestine. And remember what we said, when you have a lot of solutes in one area, water would follow. And so this is exactly what happened. So the water would follow the lactose, and you'd have the first sign, which is osmotic diarrhea. You'd always have osmotic diarrhea with people suffering from lactate deficiency. Now, that's not there. That's not it. The lactose would then continue into the large intestine. And remember, we said we had bacteria in the large intestine, right? And this bacteria, they're like best friends with lactose. Yeah. So the bacteria, once it sees lactose, it's so happy, it eats the lactose and starts fermenting, producing a lot of gases and pollutes. And part of these gases would be like bad fouling gases that have bad smell. And so a patient or a person suffering from these two signs, which are osmotic diarrhea and bad fouling smell, lactate deficiency. Okay? And also there's a test here that could be done to test if actually a person has lactate deficiency, because one of the products from lactose fermentation by bacteria is H2. And so there's the H2 breath test where they measure the amount of H2, because this H2 would then enter into the blood vessels and will diffuse into the alveoli and be excreted during expiration. So yeah, is this clear? Yeah, basically, also this slide is the same thing. I just put this diagram right here. I wanted to show you that when a lot of solutes come in one area, water would always follow and try to dilute the solution. So we already spoke about this in a normal case. Lactate enters the intestine. They get broken down into galactose and glucose using lactate dehydrogenase, and eventually they get absorbed, right? No problem. But in a patient having lactate deficiency, lactate would enter into the intestine. They cannot be broken down anymore. And instead, this lactate would accumulate like this, and the water would follow, and leading to automatic diarrhea. And also, the lactate would then move into the intestine and meet up with bacteria, best friend, and the bacteria would ferment the lactose and break and produce a lot of foul gases. And this is the reason for bad fouling smell. And we're done. Now we have some questions. But before the questions, do you guys have any questions for me?
Yeah, everything is clear.
Okay, perfect. Now let's try. I bought 4 questions because for me these are the most high yield of topics. It was 3, then I saw another one and I said no, lazim I put this question. Okay, so question number 1. Here they say a 24-year-old male graduate participates in a clinical research study on intestinal motility. Peristalsis of the small intestine. Pretty direct and straightforward question.
Is it D?
Yeah. So, yeah, you're right. So the answer is D. So we said peristalsis, you'd have contraction behind the food and relaxation after the bolus of food. See, simple questions like this, they would start with a whole clinical scenario, not really important. And at the end, it's going to be a really simple question and direct, straightforward. So there is no point of stressing out over the exam. Now, question 2. A thorough investigation of a patient with vague signs of dyspepsia reveals abnormally low levels of motility. Endoscopy confirms an absence of the migrating myoelectric complex or migrating motility complex.
C.
Further
C.
tests, mashallah, will most likely reveal an increase in which of the following. So let's see. Is it C?
Yes, because one of the main functions of the migrating complex is to wash away the bacteria as well. So if that is absent, there will be more intestinal bacteria.
Okay, perfect, Sarah. So yeah, what you said is perfect. The migrating motility complex would have wait, let so yeah, it would not increase the gastric emptying. Now you have loss of migrating motility complex, and at the same time, duodenal motility would go down. So these two are wrong. Really good. Mass movements, they don't happen here. Mass movements, some mass movements happen in the small intestine. I don't think you guys took it. It's out of your scope. Most of the mass movements happen in the large intestine. So it's unrelated completely to the migrating motility complex. And finally, swallowing, it does not even happen there. So even by elimination, you'd find that the only answer that would make sense would be intestinal bacteria. Okay. Another question, vomiting is a complex process that requires coordination of numerous components by the vomiting center located in the medulla. Which of the following occurs during the vomiting act? We already spoke about this.
Is it E?
C.
No, it cannot be A, B, or C since both sphincters are relaxed. So it's either between D
Okay.
or E. And I think it
طيب.
would be D since the intra-abdominal, intrathoracic would also increase.
Intrathoracic and what about طيب الانترا abdominal مش عاجبك ليه؟
حتى it also increase.
Yeah,
لا
so both would increase. So how would both increase?
they would both contract.
So yeah, perfect. Once the abdominal muscles contract and the diaphragm starts contracting, it would push on and like squeeze the stomach to release its product. And even in the thorax, they would squeeze and cause increased intrathoracic pressure. And eventually you'd have the vomiting act. Perfect, perfect. Now our final question. Which of the following is a likely consequence of ileal resection.
Then what does resection mean?
So yeah, resection means you're just removing that part of the ileum. You're removing the ileum completely. So here, ileal resection, you're removing the whole ileum.
Or you're removing vitamin B12 deficiency.
Okay, good. Why?
Because the ileum is the main site for vitamin B12 absorption. So without that, we would be having a deficiency in B12.
Yeah, perfect. So this is actually a pretty simple and direct question. Probably in the exam, it would be a whole case scenario about someone having inflammation in the ileum, and you have resection of the ileum, and he presented with this, and that, headache, shortness of breath, whatever. And you think he would go everywhere, and then the question would be, which of the following is likely consequence of the ileal resection? That's the last question. So pretty simple stuff. They won't really make it difficult in you, inshallah. As long as you know the basic concepts, everything is going to be simple. Any questions? Thank you very much. Please do scan this code for feedback. And yeah, if there is any question, please do say. We're done with everything.